Welcome back to another day, another battle in aerobic respiration. Today's topic is the final stage of that process, oxidative phosphorylation. Our molecule of the moment for this lecture is a little bit different. You have to tell me what it is based off the following clues. According to the World Health Organization, obesity is considered to be a pandemic, and it claims 2.8 million lives every year. There are significant medical and cultural pressures for society to reduce body mass and adipose tissue. This is what we usually mean when we say lose weight. So my question to you is, when someone loses weight, where does the mass go and as which compounds? You'll have to consult the biochemical pathways in respiration to answer this question. The learning goals for today's lecture begins with describing the roles and functions of the major components in the electron transport system. We will then explain how redox reactions facilitate the formation of this proton-motive force that ultimately drives the synthesis of ATP. And then we will be doing a bunch of accounting. We will account the flux of key substrates such as ATP and protons in the production of this cellular energy. Like the citric acid cycle, oxidative phosphorylation occurs within the mitochondrion, and I think it's worth if we spend a little bit of time dissecting the anatomy of this key organelle. Every mitochondrion is going to consist ultimately of two membranes. There's going to be a smaller outer membrane and almost paradoxically, there's going to be a larger inner membrane. Yes, it is bigger on the inside, and that is not a typo or a mistake. It's actually by design. If you take a look at this cartoon over here, you might be able to see what I'm talking about. This outer membrane is drawn in red, and in orange and yellow, we can see that this larger inner membrane is experiencing all these wrinkles and convolutions that are required to cram this larger structure inside of the smaller one. And the word for all of these wrinkles and convolutions is called cristae. And cristae are very important because we're ultimately increasing the surface area of this larger inner membrane. And that's important because it's the location of the electron transfer chain. It is going to be the site of this ATP making chemistry. Now, what's held within that inner membrane, that space is called the matrix, and that's the location of the citric acid cycle. So, that's still important, but it's, physically distinct from the inner membrane that encloses it. I just want to make that distinction moving forward. Now, it's been said to the point of cliche that the mitochondria is the powerhouse of the cell, and yes, it does have an important catabolic role. However, I want to mention here that mitochondria have many roles in the cell, and it's frankly an insult, I think, to reduce it to just it being the furnace of our beings. In addition to being the center of aerobic respiration, mitochondria are also key apoptotic sensors. I've talked quite a bit on how cancer has many ways of arising within glycolytic biochemistry. Well, the mitochondria to a degree is aware of this, and if things start becoming biochemically cancerous, it can release certain signals to the cell that says, hey, respiration is starting to go downhill, it might be a good idea to shut things down. Now, in addition to all of that, the mitochondria also has very important roles in anabolism. It will aid fatty acid synthesis and breakdown. We'll talk more about that later. It's also going to be the site of parts of gluconeogenesis. Pyruvate carboxylase, PEPCK, the two enzymes that constitute the first bypass of gluconeogenesis, they're found in the mitochondria. It's also the site of heme and steroid synthesis. And it does really so much more than that, but we frankly don't have time to go into the details. And all of this fantastic chemistry that it does on its own, thanks to its own set of DNA and ribosomes that are contained within this organelle. So like I said, reducing it to a power plant, I think, does it quite a bit of disservice. After all, there's a theory that the mitochondrion itself used to be an entire organism just a mere couple billion years ago. But back onto the main fare, aerobic respiration. I've said before that glycolysis is inefficient. It only is going to release 5% of the chemical energy that is contained within a glucose molecule, about 146 kilojoules per mole. To fully get the 100% of the energy out of that glucose molecule, almost negative 3,000 kilojoules per mole, we need to fully oxidize that glucose, not into a pyruvate molecule, but into instead 6 equivalents of CO2 and 6 equivalents of H2O. By completely stripping every carbon in this glucose molecule of its hydride content, can we extract the maximum amount of chemical energy. And when we extract that energy, we drop it off into our energy carrier currency molecules, namely NADH and FADH2, which we've also been generating in the background throughout this entire respiratory pathway. So the goal of oxidative phosphorylation is to oxidize not only glucose, but these energy carrier molecules themselves, which we will then convert into ATP equivalents. So that's the key question here. How do we do that? How is ATP synthesis coupled to glucose oxidation and NADH, FADH2 oxidation? Originally, we believed that the mitochondria made a bunch of high energy phosphoryl compounds, kind of like phosphoenol pyruvate, and we then took these unstable compounds and made them dump their phosphates onto ATP through a bunch of substrate level phosphorylation events. The problem with this theory was that we never found such a high energy phosphorylated intermediate within the mitochondrion. So ultimately, this hypothesis was proven wrong. In reality, the mitochondria, like I said, uses the chemical energy in these NADH and FADH2 carriers to generate something that's known as a proton gradient within the intermembrane space. that tiny gap between the inner and outer membranes. So what really happens is that our NADH and FADH2 are going to provide some redox energy and power on special proton pumps. And what these proton pumps will do is relocate protons from a low concentration matrix to a high concentration intramembrane space. It's going to make it acidic relative to the matrix. And then, in a process known as chemiosmosis, we are going to have controlled diffusion of those protons back down this concentration gradient through special ATP synthesis machinery. This flow of protons through the ATP synthase complex is going to generate a form of energy, and it's not unlike a water wheel. Just like how the flow of river water down this paddle generates this rotational force in this wheel, similarly, the protons flowing through this channel are going to churn the gears necessary to take ADP and PI and shove them together into ATP. And it will do so spontaneously. The next question is, though, how does NADH and FADH2 generate this proton gradient through this electron transport chain, or ETC for short? And this is actually one of my favorite things about biochemistry. Within the inner mitochondrial membrane, there are embedded proteins that will accept the electrons stolen from glucose and contained within these energy carrier molecules. And the act of taking these electrons and passing them down a conveyor belt of redox proteins is going to generate something that's called the wireless current. That inner membrane is kind of like a live wire, and each time an electron passes through, it's going to power on and turn on the proton pump functionality of that membrane protein. And that's going to provide the energy needed to actively transport protons into the intermembrane space, therefore decreasing the pH of that zone. That's nice and all, but where do these electrons eventually go? After running this circuit through the inner membrane, electrons need to be eventually removed from the ETC by reducing a terminal acceptor molecule that then diffuses away. In most cases, that molecule is oxygen, which we then reduce into water. Removing the electrons we dump into the electron transport chain with the oxygen that we breathe is crucial because that allows us to keep this cycle going. The ETC components are beautiful beasts, and I've summarized their functionalities in the table below. Essentially, electrons enter this chain at one of two junctions, either complex 1 or complex 2. And I'm going to walk you through how this entire electron transfer chain works step by step. Let's say you're an NADH molecule and you're trying to dump your electrons onto the chain. Your entry point is going to be complex 1. which is also known as NADH dehydrogenase. The goal of this enzyme is to oxidize NADH, remove the electrons it carries, and dump it into the electron transfer chain. And when you oxidize a molecule, another one needs to be reduced. It needs to receive those electrons. And the recipient of NADH's electrons is going to be a molecule called coenzyme Q, or Q for short. This Q molecule is a hydrophobic small molecule that basically surfs through the inner membrane of the mitochondrion. And that is what will ultimately ferry the electrons on to the next step of the chain. But more on coenzyme Q later. And while this redox reaction occurs, NADH dehydrogenase will simultaneously pump protons into the intermembrane space. And that is how NADH has its electrons enter the freeway, if you will. So what about the electrons in FADH2? Their entry point is going to be complex 2, aka succinate dehydrogenase. That is the same succinate dehydrogenase of the citric acid cycle, so it's going to moonlight between these two pathways. When complex 2 oxidizes succinate in the citric acid cycle, it's going to, in turn, reduce an equivalent of FAD into FADH2. And then very quickly after that, that FADH2 that we just made is going to be oxidized again. And its oxidation is going to drive the reduction of coenzyme Q. And once we supercharge Q with electrons, it's going to swim off into the Q pool and contribute to that wireless current flowing through the inner membrane. However, unlike complex 1, there is no proton pumping action going on here. It's just pure redox. From this point, the electrons that we've loaded onto the ETC via these charged up coenzyme Q molecules will next swim along to the next station, complex 3, which is another proton pump, and its name is also ubiquinone cytochrome C oxidoreductase. Its job is to take these charged up Q molecules swimming about, take off the electrons we gave them, and relay those electrons to the next electron carrier in the electron transfer chain. And that's going to be cytochrome C. This redox reaction is also going to power another proton pump, further strengthening the proton-motive force we're generating. I want you to pay attention though, because the substrate to this enzyme complex is reduced Q only. This means that electrons flow from complex 1 and complex 2 into complex 3. I find that this is something that students often struggle with. You would think that electrons would hop from complex 1 to 2 to 3, but that's not the case. If I were to map out the electron flow as a schematic, it would look something like this. We would have electrons from complex 1 and complex 2 converge into complex 3. And then it sends its electrons on to complex 4. NADH is the electron donor for complex 1. FADH2 is the donor for complex 2. And both 1 and 2 contribute Q, which travel on to complex 3. Now, the electron carrier that relays these electrons from complex 3 onward is cytochrome C, shown over there. This is a mobile redox protein whose job is to transfer those electrons to the final complex in the electron transport chain, which is complex 4, aka cytochrome oxidase. And as its name suggests, its job is to oxidize cytochrome C, take off its electrons. Well, where do we put those electrons next? We've run out of partners to pass these electrons onto. So it's time to dump them. And we're going to dump them onto a dioxygen molecule and reduce it into water. This effectively removes the electrons from the electron transfer chain. And I know this sounds like a waste, but that's actually a good thing. It's going to help ensure that the electron traffic keeps moving forward, so these complexes can continuously pump protons into the intermembrane space. And oh look, while we do that offloading, we're going to do a final instance of proton pumping into the intermembrane space. So altogether, our proton pumping steps occurs in complex 1, complex 3, and complex 4. 1, 3, and 4. And we want to keep these pumps going because those protons will ultimately provide the energy required to make ATP. Now, I want you to take note that the ATP synthase complex is technically not part of the electron transfer chain. It ends with cytochrome oxidase, complex 4. That's because the chemistry involved with ATP synthase is ultimately not a redox reaction. But all of these are. A crucial aspect to the electron transfer chain is redox reactions. And each redox reaction can be thought of as a pair of reductive and oxidative half reactions. Something's going to be gaining electrons and something's going to be losing electrons. And each of those half reactions is going to occur with a specific potential. And electrons will transfer from redox couples with low potential to high potential spontaneously. So what does that mean in plain English? Let's look at this table. I know it's really intimidating, but we're going to break it down bit by bit. And let's try to find something in here that looks familiar to latch on to. The electron transfer chain pretty much begins with NADH, right? So we can find it on this table over here, and by convention, we always write half reactions as a reduction, just to keep things consistent and organized. So this half reaction is going to be NAD plus a proton plus 2 electrons. Combine them all together, we get reduced NADH. This is a reduction. And its potential to get reduced is shown over here, negative 0.32 volts. Well, what does that mean? Is that a high potential? Is that a low potential? Well, that number, believe it or not, doesn't really matter that much on its own. What it matters is what we compare it to. So, what redox reactions does NADH participate in? Well, it's going to do a redox reaction with complex 1, also known as NADH dehydrogenase. When that enzyme accepts 2 electrons, we're going to get a reduced version of that enzyme. And its potential to get reduced is negative 0.3 volts. Again, what does that mean? On its own, not that much. The important thing, though, is that negative 0.3 is a bigger number than negative 0.32. And what this table is trying to describe is that the greater the reduction potential, the greater the propensity of that species is to accept those electrons. Electrons are going to flow from carriers of lower E value to carriers with a higher E value or potential. Said another way, species that are high on this table pass electrons to species below it. Electrons flow down this way, spontaneously. And hence, the spontaneous reaction is for the electrons to flow from NADH to the first stop on our electron transfer chain, and not the reverse, which would be disastrous. This is important because what it's showing here mathematically is that the electron transfer chain has a specific directionality. It is spontaneous to go on it, not off of it, at least from the perspective of NADH. And to prove that mathematically, we can refer to this equation down here, which is called the Nernst equation. And for this course, I really don't expect you to manipulate it and make any robust calculations. That's really more of a task you'll do in analytical chemistry. But it's going to relate these redox processes to something that you understand, which is delta G. Is it going to be spontaneous or not? Positive or negative? Let's break down the Nernst equation. It is going to boil down to delta G equals negative N, which is the number of electrons being transferred, which you can get from these coefficients over there. This fancy F, which is the Faraday constant, doesn't really matter what it is, it's just a big number. And something that's called the E naught cell, the standard potential of the cell, which is the difference between the reduction potentials of your electron acceptor and your electron donor. Said another way, your electron acceptor is going to be the oxidant. Your electron donor is going to be the reductant. Complex 1 accepts electrons from NADH. So we're going to plug in negative 0.3 over here, minus negative 0.32. What does that equal? Positive 0.02, which we're going to multiply by negative NF, a negative term. So that's going to equal a negative number, because a positive times a negative is a negative, so delta G equals a negative number. Therefore, it's spontaneous. And if you follow through this table, what we're going to observe is that the next step in the electron transfer chain, which is going to be reduction of Q by NADH dehydrogenase, that's going to have a higher reduction potential. What's the species in the electron transfer chain that accepts electrons from Q? It's going to be complex 3. That passes along to cytochrome C, and then that passes along to complex 4. And where does the chain end? With terminal oxygen reduction. And oh look, oxygen's reduction potential is a whopping 0.817 volts. It's got the highest reduction potential in this chain, and that's why oxygen is so good at oxidizing things and receiving electrons. Now we examine each complex in detail. The full name for complex 1 is NADH ubiquinone oxidoreductase, and it's a huge L-shaped enzyme that contains 40 polypeptide segments to it. It's got some crazy quaternary structure. And the arm portion that sticks out into the matrix has the job of finding, binding, and oxidizing NADH. The electrons that we take from NADH get relayed to an FMN group, flavin mononucleotide. And then from there, we're going to pass those 2 electrons to a chain of iron sulfur clusters, specifically 8 of them, 8 iron sulfur clusters. And it's going to ping pong all the way up until we get to this special binding pocket right over there against the inner side of the inner mitochondrial membrane. So floating within the inner mitochondrial membrane is a pool of molecules called ubiquinone. And you can see their molecular structures down here. We also call this coenzyme Q, or Q for short. Q is a very hydrophobic molecule. It has a hydrocarbon tail that will help anchor it into the membrane. And it possesses a quinone head that is redox active. When Q is oxidized, ubiquinone, it's going to exist as an anti-aromatic quinone. When we give it 2 electrons and 2 protons, we reduce those 2 ketones into alcohols, giving us QH2, or ubiquinol, which is aromatic. And this reduction occurs right over there at that Q binding site. So the electrons hop off onto coenzyme Q, forming ubiquinone, which undocks and swims away along the inner mitochondrial membrane. And while these crazy redox reactions happen, these 4 subcomponents that are in the membrane arm are going to pump 1, 2, 3, 4 equivalents of protons. 4 protons pumped per NADH vectorially. What does that word mean? This is a fancy way of saying that the enzyme always relocates those protons from the negative side, or N side, to the positive side, P side. And we call it that because it has an abundance of positively charged protons. And we can see that being represented in this vectorial equation. Where we have 5 protons on the N side and end up with 4 on the P side, shown over there. Complex 2, succinate dehydrogenase, is again special because it moonlights in oxidative phosphorylation and the citric acid cycle, and this complex directly couples these two processes. Just like with complex 1, its first active site is going to be on an arm protruding into the mitochondrial matrix, where it will oxidize succinates from the citric acid cycle into fumarate. And while it oxidizes succinate, it's going to be reducing FAD into FADH2. Now, the FADH2 that gets generated is immediately reoxidized within that enzyme. No diffusion required, like in the case of NADH. FADH2 is reoxidized, and those electrons, those 2 electrons that we gave it, are passed along onto a chain of iron-sulfur clusters. Similar to what we observed with complex 1. Those electrons are going to ping pong all the way up until we reach a Q binding site, where the final iron-sulfur cluster will reduce Q into QH2. And that QH2 will undock and diffuse out into the inner membrane of the mitochondrion. I'm going to re-emphasize that complex 2 doesn't accept electrons from complex 1. It accepts electrons from succinates, and by extension, FADH2. And for this reason, FADH2 is considered to be not as energetically valuable. Complex 1, if you remember, has this giant membrane arm that will pump 4 equivalents of protons for every NADH oxidized. There is no pump component to complex 2. Therefore, FADH2 contributes less to the proton-motive force than NADH does. And I'll show you how the math works out later, but generally, 1 NADH is going to net you 2.5 ATP molecules, whereas 1 FADH2 is only worth 1.5. Complex 3, formerly known as ubiquinone cytochrome C oxidoreductase, is where we converge the QH2s made from both complex 1 and complex 2 simultaneously. It's the convergent sites. For 1 and 2. And here we are going to transfer the electrons from Q onto a different mobile carrier called cytochrome C. Coenzyme Q is a small molecule that floats within this inner membrane. Cytochrome C is a small protein that likes to ride along top of that inner membrane. And while we relay these electrons between these 2 carriers, we pump another 4 protons vectorially. Let's now go through the mechanism for complex 3, which is also known as the Q cycle. Fair warning, the Q cycle is rather involved. So my recommendation is to take it slow, don't get frustrated, and there's no shame in just going back to this point and watching through the video again, because it's frankly a lot to digest in one sitting. But as always, we'll first discuss the big picture, the forest, before we get into the weeds. The goal of the Q cycle is to regenerate Q from all this QH2 that we made. So we can recycle it back to complexes 1 and 2, so we can keep this electron traffic going. While we reoxidize QH2 back to Q, we want to reduce cytochrome C, which is the next electron carrier. The problem is that Q is a 2-electron carrier, whereas cytochrome C is a 1-electron carrier. So this requires complex 3 to organize this complicated conversion in a series of 10 reactions, which we're going to outline below. Stage 1 will be the first 5, and then in the next slide, you'll see the next 5. Now let's begin. Step 1 of the Q cycle is 2 binding events. One QH2 molecule, which is reduced, will bind what's known as the P site. The P site is over here. Let's use a different color to highlight it. We use purple. Simultaneously, an oxidized Q will bind the N site. And let's use blue. All right, that's not so bad, right? Step 2, QH2, which is in the top, is going to transfer 1 electron to Q. Which is on the bottom. And I'll try to keep the color coordination consistent. So I'm going to paint the path of electrons down that way. Giving 1 electron, 2Q converts it into semiquinone, Q minus dot. And I'll give that a green circle. But that's not all. When we lose an electron from QH2, it also becomes Q dot minus, semiquinone. So 2 semiquinones are made, one in the top position and the other in the bottom. And I will again highlight that. Now, step 3. the top semiquinone, which is in the purple place. That's the one that used to be QH2. That top semiquinone is going to transfer another electron. It will give its second electron to cytochrome C. And that I can highlight in yellow. So its electrons are going to travel up like so. Once we do that, the semiquinone that was in the top position becomes just regular Q. And let's try highlighting it with light green. It's becoming very colorful. So now here's the situation. It's step 4. The E site releases the Q that used to be QH2. And that's because the P site, the top site, prefers QH2 over Q. At the same time, cytochrome C also diffuses away. Lastly, 2 protons that used to belong to QH2 are stripped and pumped into the intermembrane space. This is the hard part. If you can get this, the second half I think will be easier to understand. But trying to bring it all together in one sentence or 2 sentences, we're going to remove the electrons one at a time from the QH2 in the top. One's going to go down into a Q, the other's going to go up into cytochrome C. And these 2 protons that get left behind get released into the P side, the intermembrane space. This constitutes stage 1 of the Q cycle. Stage 2 is going to be all about dealing with this semiquinone in the N site. Continuing on with the second half of the Q cycle, step 6 is going to look familiar. We will have a second instance of reduced ubiquinone, QH2, binding the P sites right over there. That's what we did essentially in step 1. Step 7 will also be familiar. We will have two individual single electron transfer events occurring from this newly docked QH2. So that QH2 and the top P site will transfer one electron down to that semiquinone radical that we did not forget about. While the semiquinone finds two protons from the matrix side. We see that over here. That second electron comes in and merges with two H pluses, yielding QH2. Step 8. The bottom, newly formed QH2 does not like to be bound to the end site, so it's going to diffuse from it. And leave, heading into the inner membrane. Step 9. The top semiquinone, which used to be QH2, it's now a Q dot, transfers the second electron to a new cytochrome C. Right over there. And in doing so, the last step, final step of the Q cycle, two protons are stripped, and pumped again, right over there. So in a nutshell, we have altogether step 6, which is binding of QH2 in that top slot. Step 7 is the first electron transfer event downwards to that semiquinone. Step 8, we pick up two protons from the matrix and form a brand new QH2, which is then going to leave. Step 9, our second and final electron transfer event from the Q semiquinone to the cytochrome C. And once we lose that final electron, we're left with just regular old Q. The Q also leads into the inner membrane, and the protons that get stripped from it released into the intermembrane space. And that is how we do this very complicated transfer while pumping protons from this side of the membrane onto Q, which winds up right over there, and then releases those protons to the intermembrane space. Pretty clever way of getting protons across that very hydrophobic membrane. The last stop, complex 4, cytochrome oxidase, is a large and mysterious enzyme. Its job is to transfer electrons from cytochrome C, that's the shuttle that we just charged up in our previous step, and it's going to dump those onto a molecular oxygen molecule, reducing it to water. This is going to be the final stop of the ETC. This protein is highly conserved, which means that its sequence and structure does not vary tremendously from organism to organism. It's going to look quite similar in a bacterium compared to, let's say, a human cell, which means that it does its job pretty well. That being said, the mechanism is poorly understood. But we do know it achieves the following. That cytochrome C molecule that we just loaded up at complex 3 is going to diffuse over to complex 2 and dock over here on the P side. And from there, it's going to dump electrons into a copper cofactor pair. From there, it's a short hop to this pair of hemes near this iron copper center. And that is going to be the site of oxygen reduction. Oxygen will be the terminal electron acceptor in the electron transfer chain. We're going to offload these electrons as it diffuses through the membrane into the protein site, and then it's going to accept those electrons we loaded into the iron copper center. And when we do that, it is going to merge with protons from the N side, the matrix side, and form water. While all of this happens, the complex will be pumping protons vectorially, specifically 4 of them. 4 protons pumped. And we can see the equation down over here. 1 equivalent of O2 or 2 equivalents of oxygen atoms will yield 2 water molecules and 4 protons being forced through. Now at this stage, I want you to consider something. Why do you think complex 4 insists on binding cytochrome C and not coenzyme Q and not NADH or FADH2? There's no binding pocket for either of these electron shuttles. No binding pocket. And that is intentional. See how wise it is to change up carriers throughout the electron transfer chain sequence. This is going to create a specific route for the electrons to follow that will maximize the protons pumped. If Q can dump the line to complex 4 and skip complex 3, that's going to short the circuit. We're going to lose out on the protons that are pumpable through the activity of complex 3. The same goes for NADH and FADH2. If they can bind here and drop off their electrons, we're skipping the pumps in complex 1 and complex 3. So yeah, it's kind of annoying to switch up these carriers, but this is actually going to maximize our yield of protons winding up in the intermembrane space. And that's really important for the final step of this entire respiratory process, ATP synthesis. Now, before we get into that, let's take a step backwards and examine the electron transfer chain at a distance. We have electrons entering this highway at this entrance ramp, complex 1, and at this entrance ramp, complex 2. Let's put a pin in complex 2 for the next couple of minutes, and I want you to just focus on the path of electrons taken from one equivalent of NADH. All right, and let's ask ourselves, how many protons do we get pumped into the intermembrane space as a consequence of this? Well, let's start at complex 1, and that's going to pump in 4 protons by oxidizing NADH and transferring its electrons onto Q. The ensuing QH2 will diffuse over to complex 3, and that's going to trigger a second pump, which will give us another 4 protons. And QH2's electrons get transferred to cytochrome C. That moves on to complex 4, where we're going to do our final electron transfer event, where we dump those electrons onto an oxygen molecule, giving us water. And while we do that, we pump a final instance of 2 protons into the intermembrane space. Now, this is a little confusing because I know in the last slide I said 4, and that's because that's how many protons you get for doing it twice for 2 oxygen atoms or 1 O2 molecule. Since we're just focusing on the journey of a single equivalent of NADH, it only needs technically half of an O2, 1 oxygen atom. And that's because 1 NADH is equal to 2 electrons, and 1 oxygen atom has 6 electrons, and the 2 we get from NADH is all you need to make water. I've shown those here as X's instead of dots. So there you go. It takes half of an O2 to burn 1 NADH. Now, what's the key takeaway here? What I want you to know is 1 NADH equals 10 protons pumped. Said another way, every time we oxidize an NADH molecule, we're going to migrate or actively transport 4 protons into the P side. If you don't enjoy working with this equation with 1/2 of an O2 molecule, you can just multiply everything by 2, so we're dealing with integer reaction coefficients, and then you get, in terms of products, 20 protons for every 2 NADHs oxidized to NAD+, and then that will, of course, net you 2 H2O molecules formed at the very, very end. In a nutshell, what we are doing with the free energy in that glucose molecule is converting it to potential energy in the form of an electrochemical potential existing as this proton gradient, where we have a lot of protons on this side and not that many on the matrix side. This potential energy is what ultimately will drive the proton-motive force powering ATP synthase. The universe wants to even out this concentration gradient, and we're going to capitalize on that over here. So that is how we exploit our proton gradients. Really quickly, I'm going to point out to you this question: why is FADH2 not a part of the vectorial equation? Well, there's no pumping action at complex 2. We still get money out of it in the form of 6 protons, because we get to trip up this pump at complex 3 and the pump at complex 4, it's just not worth the same. ATP synthase is what this whole story is about. This protein is a channel, essentially, that will bleed the protons we spent all that effort pumping back down the gradients from the P side to the N side. And ATP synthase has many important constituents that we can divide into two main components. The F1 component protrudes into the mitochondrial matrix, the so-called N side, and this is the site that actually catalyzes ATP synthesis. And it's going to be connected to this rotating F0 subunit that's housed within the membrane. And as the protons enter and flow through it, that's going to cause this arm connected to F0 to churn. This is the arm over there. It's going to churn and spin and agitate all of these enzymes below in the F1 domain. And that's going to activate them. So the key takeaway here is that ATP synthase is a molecular motor. It's a moving, churning mechanical beast. And what's generating this literal rotational motion, seen in this GIF, is the proton flow. That's what causes the gears to churn. And that's going to provide us energy to do a really difficult reaction, namely the conversion of ADP and PI into ATP. Let's unpack the anatomy of ATP synthase in a little more detail. I'm telling you it's a molecular motor. Well, what is a motor? A motor is a machine whose job is to interconvert mechanical energy and some other kind of energy. And it's going to consist of a stationary portion called the stator, working together with a rotating portion called the rotor. The stator domain of ATP synthase consists of a protein arm that anchors the proton channel to the catalytic portion of ATP synthase below. And the inner membrane in which it is docked to, I'll draw as such. And what I want you to understand is that the proton channel running through that inner membrane is not a straight tube. It is actually two half channels. What do I mean by that? There's an entry port over here where the protons flow in, and they are going to enter essentially the rotor. They're not going to flow directly into the matrix side. And then when they're done rotating the rotor, they are going to come back to the exit port. So you can see there's two half channels. So let's follow that proton that's falling through. It's going to enter, and where does it go? Bam, to the rotor. The rotor consists of 10 C10 proteins, these columnar blue entities, and these are essentially the gears that are being rotated by these protons. The proton will dock one of those C10 units and induce a conformational change that's going to prompt it to rotate over 1/10 of the way around the carousel, so to speak. So 10 protons are going to flow through, and that's going to induce stepwise rotation of this rotor domain. And once that proton completes a circuit, it's going to get dropped off to the bottom half channel, where it will diffuse out into the matrix. Now, while the C10 unit rotates, it's not just rotating on its own in a vacuum, it will also be churning this gamma paddle attached to it, which is extending below the inner membrane. And it's actually dipping into that headpiece. The paddle, as it rotates, is going to stir the pot of the catalytic headpiece, causing these enzymes to change into 3 different conformations. Loose, which is going to bind ADP plus PI, and then it's going to turn a little bit more and rearrange its shape into the tight configuration. That's going to squeeze them into ATP, which is very hard to do. So we need that little bit of mechanical energy to do it. And then twist a little bit more into the open configuration, which releases the product. And then we rinse and repeat. So this is pretty cool. What we're essentially doing here is converting potential energy in the form of an electrochemical gradient into mechanical energy, as seen with the rotation of C10 and the associated gamma paddle. And that gamma paddle's mechanical energy is being transferred and converted into chemical energy, catalysis, formation of ATP. Let's go through the rotation-dependent synthesis of ATP again, because it is nothing short of elegant. We also call this, by the way, rotational catalysis. In the ATP synthase headpiece, we have these subunits called alpha and beta, shown as these pink and green ovals respectively, and they're arranged as a heterotrimer. The alpha subunits are structural. They help catalysis happen, but they don't actually do it directly. It's the beta subunits, the green ones, that shine and do catalysis. And what we're going to do is visualize their dynamic conformational changes by staring down the barrel of the headpiece. So we're going to look at this from the perspective of the gamma paddle, so to speak, dipping down and churning up these alpha beta heterotrimers. The gamma paddle, if you noticed, shown over here as this orange irregular blob, it's not just like a rod. It has a very weird shape. It's kind of like a wedge, or they say it's triangular, but doesn't look like a triangle to me. Anyway, depending on the orientation of this weird shape with respect to the beta subunits, that's going to dictate its conformation and what kind of functionality it can achieve in that moment. So let's begin in the following states. When the paddle points away from a given beta subunit, it's going to have that loose configuration where we will have the flexibility to bind ADP and inorganic phosphate through induced fit. Let's rotate the paddle 120 degrees. Now the paddle is facing sideways with respect to that beta subunit. It is going to force it to tighten onto that substrate. And that squeeze of the grip is going to impart the mechanical energy required, so to speak, to force the PI onto the ADP, giving us ATP. Hooray, we've made it. Now we need to loosen our grip again, open up, and release our product. So the paddle is going to turn a third time, the final 120-degree rotation. The paddle's facing in, and it's kind of like when you release ice cubes from the tray, you have to like protrude your thumbs into the bottom of it to like pop it out. And that's essentially what happens. Protrudes inward, and we're going to open it up and release our ATP, and that's going to let us go back to square one and repeat the process. And this process is quite fast. It happens about 100 times every second, and there's 3 beta subunits all operating at different stages. It works just like a car engine when you think about it. When it's open, that's going to be fuel intake. When it tightens, now you've got ignition. And then when it opens again, you get to have exhaust. And then you repeat that cycle. Let's wrap all this up with a little bit of accounting. We already learned so far that one NADH molecule is ultimately quote unquote worth 10 protons pumped into the intramembrane space. We achieve this through the combined pumping activities of complexes 1, 3, and 4. FADH2, on the other hand, is worth a little bit less. We only pump 6 protons into the intermembrane space, and that's because we're bypassing the 4 protons pumped normally in complex 1. So the next question that you may be wondering is, how do we convert these values into ATPs made? That's ultimately the goal of aerobic respiration, is it not? To make ATP? Well, the answer to that question is a little bit complicated. One NADH molecule is ultimately going to be worth 2.5 ATPs generated. And on the flip side, since FADH2 only gives us 60% of the protons that one NADH does, and these protons are ultimately what drives ATP synthesis, we get 60% of 2.5 ATP units, which is 1.5 ATPs. Now, where do we get these weird numbers from? And I should first state that these exact values are estimates and they are debated. Some scientists think that these numbers are bigger and some think that they are smaller. But we get non-integer values because ultimately it has to do with how ATP synthase has 3 catalytic sites, and they all cycle with every 10 protons that go through it. So remember that the rotor C10 has 10 subunits, and 10 protons are going to cause 1 complete rotation. There are 3 beta sites, each with the potential of making an ATP. So 3 ATP equals 10 protons. Now you might be thinking, well, these numbers still don't add up, because if we take 10 protons and we divide that by 3 ATPs, wouldn't that be 3.3 repeating? Well, we forgot about something. What about the phosphates? We have to get these from somewhere. And this phosphate translocase symporter that's also docked within the inner membrane is going to exploit this proton gradient to pump in phosphates to fuel the beta-catalyzed ATP synthesis reaction. So inward motion of the proton is going to drive inward motion of inorganic phosphates. Said another way, every phosphate that we put in to this reaction is going to cost us an additional proton. So all in total, it actually costs around 4 protons to generate 1 equivalent of ATP. More accurately, it's going to be closer to 4.3 protons. Per ATP. How do we connect this back to NADH? Well, 1 NADH is tantamount to 10 protons. This in turn is tantamount to 4.3 protons per ATP. And how many ATPs can we get out of that? X ATP. Well, you just solve for x, and you divide both sides by 4.3. 10 divided by 4.3 is going to be equal to about 2.33 ATPs. And we round this up to 2.5 just to make the math look nice, because the rules don't matter and the points are all made up, right? The fact that we have a non-integer value is actually proof that ATP synthesis does not occur via substrate level phosphorylation. That process is a chemical and therefore stoichiometric. Rotational catalysis is biological and non-stoichiometric. As a bonus, I found this really cool GIF of the different moving parts of the electron transfer chain cooperating together. So it may be good practice if you took a minute and just tried to identify all of the molecules and events happening here. But that is indeed, in a nutshell, a very big nutshell, how aerobic respiration works. The next lecture will be about its regulation and breakdown. Until then, have a good one.